Hello, everybody, and good afternoon. First of all, I would like to thank the organizers for inviting me to give this talk and to share with you some of the activity that we do in my lab. In this work, we are collaborating with Michelle and Bruce Kendall. And this work is the vision and legacy of a very unique lady. Bruce, please.
In early 2016, at just the age of 43, Michelle was diagnosed with advanced stage ovarian cancer. She went through conventional treatment and entered into alternating sequences of remission and recurrence. In 2018, she stumbled across the then emerging literature on cannabis as a potential treatment for cancers, and quickly got to the point of realizing that the responses of cancers to cannabinoids was specific to every cancer. So she came to the question, what do I need to take to cure my cancer, ovarian cancer at this point? And some of you may remember her from CannMed 2019, when she was here with this glittery hat, with a videographer in tow, handing out bumper stickers and asking everyone that she could find, who's working on ovarian cancer. And the answer which she got was nobody. Well, so the film, she did make her film, and it was ready to go onto the film festival circuit in March of 2020, and of course that all got canceled. And then she turned, and together we turned and decided that it was going to be up to us to find the research that might cure her disease. So at that point, she made contact with Hinanit, and we entered into an agreement where we would fund the research in Hinanit's lab to look at the preclinical studies of cannabis on ovarian cancer, both with the hope of saving Michelle's life, but also looking for a cure to this terrible disease, which has a very high mortality rate because it's almost always diagnosed at advanced stages. This is the hard part. Hinanit went to work very fast, and we got some really promising early results. But by the time we could turn those results into a formulation that Michelle could use, she had already progressed to organ failure, and it was just too late for this to save her life. But before she died in September of 2021, she asked both of us to promise to keep pushing this work forward in the hopes that we might find a cure for all the other women who are afflicted with this disease. So Hinanit and I are carrying this on. It's really mostly Hinanit who is doing the work, but I'm so happy that she's my partner in this.
Thank you, Bruce. It's not easy to continue after your words. Okay, so we all know about cannabis, and we know that cannabis produces hundreds of different compounds, and that we are usually not sure what compounds are really important, and more than that, what is the preferred composition or the best composition to confer the entourage effect that Dr. Russo was talking about, And this is especially in light of the variety of different strains, cannabis strains that contains different compounds. In my lab, we ask what is the best composition of active compounds for the optimized treatment of each medical condition. And to answer that, in my lab, what we do is we extract cannabis from different strains. We separate the extracts to fractions, and by that we are able to simplify, in a way, the chemical composition and to ask the question, what compounds and what composition of compounds are working best on biological assays. Ovarian cancer, indeed, is the second most common and the most lethal gynecologic malignancy in the Western world, and very few women survive 5 years after diagnosis. There is only little is known about the activity of cannabis against ovarian cancer. Definitely the active compounds were not identified before we started our work. So first of all, what we did is we looked for different strains in order to find the best ones that are mostly active against ovarian cancer cells. We looked for cell viability of ovarian cancer cells, of several cell lines. And we were able to identify one of the strains. In this case, it is DQ, which is a high THC strain. And we found that this strain is highly active in comparison to others. Then we fractionated the extract and were able to find and to identify the actual fractions that are highly active. Following chemical analysis, we were able to determine the API, the formulation, that is highly active in terms of cytotoxic activity against ovarian cancer cells. And the formulation contained mostly THC, some CBC, and just a little bit of CBG. In order to prove that indeed this is the active composition, what we did is we then used purified phytocannabinoids in this composition, in this ratio, in order to prove that indeed this composition is the active one. And indeed, once we used phytocannabinoids only, and remember, this is originated from a fraction, which contains many more compounds rather than only this. But yet, when we re-established this composition using purified phytocannabinoids, then activity was gained as much as in the fraction. So this led us to the understanding that the actual active compounds are these ones in these certain ratios. Following, we have determined, using antagonists and inverse agonists and blockers, we determined receptors that may mediate cannabis activity, F7 diffraction, and also the standard mix SM. And we found that CB2 is mostly involved, and there was some evidences for TRPV2 as well. However, it is most reasonable that to treat patients with ovarian cancer, the treatment will not rely on cannabis only, but rather cannabis will be used as a co-treatment with another chemotherapy. And indeed, we have examined the activity of cannabis, of the fraction, and then the standard mix with different chemotherapies. We found a synergy with some of them. And in this talk today, I will focus on the PARP1 inhibitor, niraparib. Niraparib is the second line of treatment of ovarian cancer patients. What we could find is that the fraction, and not the standard mix, but the fraction itself from the plant, led to synergy with niraparib. So what we see here is sensitization of ovarian cancer cells to the PARP1 inhibitor niraparib. Then we wanted to examine the activity of the combination of the fraction and niraparib against normal cells. We were able to show that this combination was 50 times more active on cancer cells than on normal cells. These were keratinocytes in this case. And then we were able to show that the combination of cannabis fraction and niraparib was indeed active, more than niraparib only, on Michel's cells. Cells were isolated for Michel tumor and were shifted to Israel, to my lab, and indeed you can see the result. We've conducted in vivo study and conducting additional ones. Already in the first experiment, there was an advantage for the use of cannabis with niraparib. Avastin was added in this case, versus a treatment with niraparib and Avastin only. And then we wanted to get deeper and to better understand the mode of action of cannabis and niraparib against ovarian cancer in vitro. First of all, we showed that the activity is conveyed via apoptosis and not necrosis or other form of cell death, but rather, indeed, there is an induction of apoptosis by especially by the combination of niraparib and the cannabis fraction. Following, we have looked at the Wnt pathway. The Wnt pathway is deeply associated with cancer in general and ovarian cancer in particular. Cannabis, the combination of F7 of cannabis fraction and niraparib led to significant reduction in some of the components of the Wnt pathway, including the receptor, which the frizzled receptor, Fz, in the drawing here, which is essential for activation of the Wnt pathway. So cannabis, by its own, by the way, and the combination of cannabis and niraparib reduce significantly the expression of the receptor and additional components. We've also showed that the combination of cannabis and niraparib, F7 and niraparib, lead to relocation of proteins, beta-catenin in this case, again, part of the Wnt pathway, relocation to the cell membrane. This and others are very important features of the mesenchymal to epithelial transition. Mesenchymal phenotype cells are those that are related and responsible to metastasis. And these are usually resistant to chemotherapy. Once these cells were treated with F7 and niraparib, the mesenchymal features changed to epithelial features. And this is associated usually with cells that are not metastasizing and are not resistant, usually. So, indeed, we suggested that cannabis treatment of ovarian cancer cells reduce the proportion of a mesenchymal cell and increase proportion of apoptotic cells. And this is by altering the Wnt pathway. And this is all published. It was published last year in Molecules. But following, we wanted to go even deeper into the mode of action of F7 and niraparib. And to do that, what we did is we sequenced cells from 2 cell lines of ovarian cancer that were treated with the combination of F7 and niraparib. We found hundreds of different genes and several dozens of genetic pathways that were significantly altered by the F7 plus niraparib treatment in both cell lines. These biological and signaling pathways that were altered suggested some features of activity of F7 and niraparib that were verified and are still being verified in an ongoing research. And these included, for example, inhibition of cell migration and differences in cytoskeleton F-acting rearrangement, and led to a better and more comprehensive understanding of the activity of F7 and niraparib and the basis of their synergy and the ability of cannabis to synthesize cells to chemotherapy, PARP1 inhibitor in this case. So if I summarize in few words, we have started with many different compounds, many different strains, and the inability to determine what compounds are active and what compositions are the optimal ones. Using different bioassays, chemical analysis, we were able to get a formulation of API and get deeper into the targets and mode of action. These, of course, have to go through toxicity studies and additional in vivo to determine the actual activity and the ability to go into clinical trials. And of course, only successful clinical trials will prove effectivity. And then the actual formulations that were identified might serve as a base for chemovars of cannabis that will be optimal for treatment of this and other diseases. So the information on the API formulation may be translated, and I hope it will be translated in the future, not just into new cannabis-based products that will be specific and at pharmaceutical grades, but rather this information would serve as a base for breeding for new cannabis chemovars with certain activities. And with that, I would like to thank my team in the lab, and especially Nurit Shalev, which is with us today, and she is leading this research in my lab. Thank you.
Do we have time for a question? That was really interesting. It blows my mind that this specific set of cannabinoids in this ratio is suddenly so active. How specific is it? Or have you ever tested the tolerance of these kinds of specific ratios to see how close you have to get?
Absolutely, absolutely. What we did, and I did not show that here, but what we did is determine the range of ratios that we need to use in order to have synergy. So definitely, what I showed you is just an example. Rather, and it's in estimates. You can understand that 3/4 should be THC and 1/4 should be, and so on, CBC and so on. However, we carefully determined the boundaries of synergy. And by that, using these 4 components, we could draw the synergy in terms of activity and between the 4 cannabis compounds, but also with niraparib. Okay, so yeah. Thank you. Okay. Thank you.